Hey everyone, I'm Mo Kashkush. Today I want to talk about a presentation I gave a few months ago about Trodelvy and metastatic breast cancer. This information was all current as of December 2020. So let's start with a disease overview. What is metastatic breast cancer? So breast cancer usually originates in the milk ducts where abnormal and unregulated cells can grow aggressively. At this point, the disease is still considered non-invasive, but these cancer cells can actually progress past the ducts and invade the surrounding tissue and lymph nodes. Unfortunately, these cells can then enter the broader circulatory system and penetrate a distant site, forming a secondary tumor or metastasis. The metastases tend to occur in organs like the brain, lung, and liver, but can also occur in non-visceral sites such as the bone. Patients tend to present with symptoms that you would expect in these sites, so progressive neurologic and cognitive decline with an increased stroke risk in the brain, respiratory symptoms and lung failure in the lung, jaundice, abdominal pain, and fluid buildup in the liver, and severe bone pain and an increased fracture risk in the bone. Now, despite being the least common to occur, you'll see from this Kaplan-Meier curve that brain metastases, which is the red line, has the worst prognosis, and bone metastases in the blue has the best prognosis. The median patient has an overall survival of less than 1 year compared to over 2 years when you look at other sites on this chart. Not all cancer cells are the same, with different molecular markers having important prognostic and treatment implications. So when estrogen or progesterone bind to intracellular receptors, causes cancer cells to grow and divide. So the presence of either of these receptors can get categorized as a hormone receptor positive disease. As the name implies, for a human epidermal growth factor receptor, this also contributes to cancer cell growth and division even more aggressively than hormone receptors. So overexpression of this extracellular receptor is called HER2 positive disease. Finally, the subtype that I'll focus on most for today, triple negative, isn't really defined by what it is, but rather by what it isn't, meaning that it's negative for both hormone receptors and HER2 receptors. Besides being the pathologically most aggressive subtype, the historical lack of therapeutic targets is part of why this subtype has the worst prognosis, as shown on this survival curve on the left, with the median overall survival only being 10 to 13 months compared to 3 to 5 years for other subtypes. In fact, only actually 11% of patients are still alive 5 years after they get a metastatic diagnosis. So what's the overall disease burden in the US? Well, breast cancer is the leading cause of cancer in women in the US, and it's the second biggest killer, second only to lung cancer. Now, there's over 150,000 patients living with metastatic breast cancer, and this is largely made up of patients who were diagnosed early and progressed, but also about 6% of patients who received their first diagnosis at this stage. And nearly all of the more than 42,000 deaths expected from breast cancer this year will be due to metastatic disease. So lifetime exposure to both exogenous and endogenous estrogen, stage and molecular subtype at diagnosis, and having tumor suppressor gene mutations are all risk factors for developing metastatic disease. Specifically for developing triple negative disease, it's known that young Black women are at the highest risk. So the data shows that breast cancer mortality as a percentage of the US population has declined steadily since 1990. But This is not simply due to less people getting breast cancer, as suggested by improving 10-year survival curves on the left. But what's less clear is how much of this survival benefit is due to improving treatment, since widespread awareness campaigns have also caused a surge in earlier stage diagnosis, which actually also does have a positive effect on survival. So now let's talk about treating metastatic breast cancer. We won't discuss supportive care for different types of metastases or how patients with different demographics, comorbidities, or prior treatments are handled. But I at least want to mention that treatment is definitely not formulaic. Ultimately, every patient's different and requires careful consideration. With that said, the overarching principle here is to treat based on the molecular subtype. So that's really what I'll focus on. Now, for hormone receptor positive disease, it's treated with endocrine therapy, also known as hormone therapy, which basically involves either blocking estrogen at the point of synthesis with aromatase inhibitors, or blocking its effect by downregulating or modulating estrogen receptors. Now, cell cycle inhibitors like CDK4/6 can be used in combination, but never alone. In second line, it's really the same principle here, but alternating through endocrine agents that's sometimes up to 3 times before chemotherapy is even considered. You also have genome-targeted therapy more recently, such as PARP inhibitors for patients with BRCA mutations, BRCA mutations, and patients with all 3 subtypes, actually, so not just hormone receptor positive. We also have PI3 kinase inhibitors for PIK3CA mutations in patients who are hormone receptor positive. Now, in HER2 disease, we actually have multiple targeted options. First line is often pertuzumab and trastuzumab combined with a taxane chemo. And pertuzumab and trastuzumab both target HER2, but at different extracellular domains and have a synergistic effect. But it is important to note that pertuzumab is never supposed to be used as a single agent. So a second line option here is trastuzumab emtansine, also called Kadcyla or TDM1, which is an antibody drug conjugate that we'll reference again later. So third line is trastuzumab deruxtecan, which I would expect to replace the role of Kadcyla in the coming years. But we also have a chemo combination options and the option to combine with tyrosine kinase inhibitors, which act on an intracellular domain of HER2. Now, here in triple negative disease, you should see a little bit of a stark contrast here. The standard is really limited to sequential single agent chemo, meaning that we're using one agent until progression or toxicity, and then switching to the next chemo option in second line. And here we have more of the same as long as it wasn't used in first line. So the reason we use single sequential therapy is really telling when you think about what the patient's goals of therapy are. And that's because if you combine chemo in these patients up front, you actually do achieve higher tumor response rates, but it's at the expense of much higher toxicity, and it has not been shown to increase overall survival in randomized controlled trials. So it should really tell you that patients and physicians really care about longer life and quality of life. It's much more important than just the tumor response rate. So there's been 2 recent approvals, again, recent as of December 2020. And in the last 2 years, we've seen checkpoint inhibitors such as Tecentriq and Keytruda have a significant survival benefit in first-line patients when used in combination with chemotherapy. And in the third-line setting, just in April 2020, we had the approval of Trodelvy, which we're going to talk about more now. So what is Trodelvy? It is an antibody drug conjugate with three main components. The first is sacituzumab, which is a humanized monoclonal antibody directed against TROP2. TROP2 is a cell surface glycoprotein that is overexpressed in cancers, tending to be more aggressive and resistant to other therapies. The second component is govitecan, or SN38, which is the active metabolite of irinotecan and 2 to 3 orders of magnitude more potent. So this is a case where neither component is necessarily useful in isolation, with sacituzumab not being potent enough and govitecan being too toxic. But they become useful when you conjugate them together. So that kind of brings us to the third component, which is this hydrolyzable linker that conjugates the two. You'll see that the linker can attach sacituzumab at up to 8 different sites, as shown at the bottom, with that monoclonal antibody marker where it attaches to sacituzumab. And it binds via this hydrolyzable bond to govitecan up at the top, designated by that arrow. And really, it's the fact that this toxic payload can be released in a controlled fashion in low pH conditions at TROP2 expressing cells that makes this so powerful, especially when you consider the high drug antibody ratio of 7.6 compared to older generation ADCs like Kadcyla, like we mentioned before, which average about 3.5 toxic payloads per antibody. So it primarily works like this. The ADC is administered and preferentially binds to TROP2. It binds to TROP2 overexpressing cells, and it's subsequently internalized via endocytosis. The endosome becomes a highly acidic environment called the lysosome that causes the hydrolysis and release of govitecan, where it enters the nucleus and blocks topoisomerase 1. By blocking topo 1, govitecan effectively stops DNA replication required for cancer growth, and this causes apoptotic signals to be released, and it eventually causes cell death. So let's take a look at the evidence. And, it's important to remember that this is all great. In theory, everything I've shown you suggests that Trodelvy is bioplausible as a therapeutic in TROP2 overexpressing cancers, but we need to look at the actual clinical evidence to know if it works. But we do need to look at this evidence in the context that the FDA looked at it during review. This context was accelerated approval. So in life-threatening diseases with few treatment options and high unmet need, the FDA rightfully allows for expedited approval of therapies with a much lower threshold of evidence than would typically be required in gold standard regular approval. I hope that I've shown so far that metastatic triple-negative breast cancer, especially in later lines of therapy, definitely meets this criteria. But importantly, these therapies do not need to prove a survival benefit until post-marketing confirmatory trials, and often can actually gain approval in pivotal trials based on endpoints earlier than survival that are reasonably likely to predict clinical benefit. These usually amount to objective response rate and duration of response. So, what is ORR? ORR, it's the sum of the partial responses and complete responses in a clinical trial, which are measures of tumor response. PR is achieved in a patient whose tumor is reduced by over 30% in the sum of diameters, and CR is generally achieved when lesions are completely eliminated. So the trial itself for Trodelvy was a phase 1/2 basket trial that enrolled patients with many different types of metastatic epithelial cancers that had relapsed after at least one standard regimen. Trodelvy was partially approved on the basis of subgroup efficacy analysis, where patients had to have triple-negative breast cancer They had to have received the 10 milligram per kilogram dose of Trodelvy, and they had to have progressed on at least 2 prior lines of therapy. So these 108 patients made up the efficacy population, while the larger basket of 420 patients who received at least one dose for other cancers were analyzed for safety. Notably, this was a single arm analysis, so we can really only describe the outcomes rather than compare them to another intervention. Patients received an IV infusion on days 1 and 8 of a 21-day cycle, but dose reductions and interruptions were allowed if adverse effects occurred. As the basis of approval and primary endpoint, we'll focus more on the ORR in this study, but we will look at survival analysis in the confirmatory trial. So some things that stand out in the efficacy patient population were the underrepresentation of Black patients despite their relative propensity to develop the disease, as well as the general absence of patients with brain metastases. Patients were also excluded if they had a poor performance status. Otherwise, I do think that this patient population was adequately represented by age, number of prior treatments, and frequency of visceral metastases. You can see from the waterfall plot at the top that 1/3 of patients had an objective response, defined by the amount of patients falling below that -30% cutoff line, or that 30% reduction cutoff line. 3 of these patients did achieve a complete response. Now, the swimmer plot on the bottom left shows that patients had a median time to response of 2 months, and the response lasted for 7.7 months before eventually progressing or dying. I've reported the median PFS and OS, and that's 5.5 and 13 months, but I will hold off on this a little bit longer until we look at the more robust confirmatory trial. So the side effect profile was shown to be substantial, with every patient having at least one adverse effect and 72% of patients having grade 3 or 4 effects. The ones listed on the right are in descending order by frequency, and they all occurred in at least 5% of patients. Dose reductions and interruptions were relatively common, but only 2% of patients actually discontinued. The 4 deaths that occurred near a dosing interval were all considered to be related to the disease actually progressing rather than due to the drug. And, the most notable effects here are considered manageable, but still severe enough to receive black box warnings in the case of neutropenia and diarrhea, as well as a general warning to monitor patients with UGT1A1 liver enzyme mutations because of their reduced ability to clear govitecan. And they're shown to be at an increased risk of having adverse effects because they can't clear govitecan as quickly. Ultimately, this acceptable safety profile relative to the severity of the disease, as well as the historically poor efficacy of comparable third-line chemo agents, was enough for the FDA to grant accelerated approval of Trodelvy in April. As I mentioned previously, however, this approval is contingent on the clinical benefit shown in confirmatory trials. So just reading the notice from the FDA here, Trodelvy was approved for the treatment of adult patients with metastatic triple-negative breast cancer who have received at least 2 prior therapies for metastatic disease. And on the package insert, it states, this indication is approved under accelerated approval based on tumor response rate and duration of response. Continued approval for this indication may be contingent upon verification and description of clinical benefit in confirmatory trials. So what about the confirmatory trial? Although the full trial results haven't been published, I think it's still worth taking a look at the abstract presented at ESMO in September 2020. The ASCENT phase 3 trial enrolled 468 patients, although it would have been 529 if they didn't exclude the patients with brain metastases. Patients were randomized 1-to-1 to Trodelvy at the same dose regimen as we previously talked about, but this time they were compared to single-agent chemo regimens of the physician's choice, which were one of the 4 agents listed at the bottom. So capecitabine, eribulin, vinorelbine, and gemcitabine. The primary endpoint was progression-free survival, but overall survival, objective response rate, and safety were also examined as secondary endpoints. So in this trial, we saw a clinically relevant and statistically significant improvement in PFS, extending by over 4 months and reducing risk of progression by 59%, as well as overall survival improving by 5.4 months and reducing death risk by 52%. And a 7-fold increase in ORR, including 10 complete responses compared to only 2 in the control arm. These benefits were so large that the trial actually was stopped early due to a clear efficacy signal. Notably, the adverse drug events were considerably more prevalent in terms of neutropenia and diarrhea. Unfortunately, as this is only the abstract that I saw, that's all I can really say about this study, but it's definitely promising. Notably, this is the first antibody drug conjugate to show a survival benefit in heavily pretreated metastatic triple-negative breast cancer. So on top of this sort of journal club and disease state overview I've given, I do have a SWOT analysis and I guess sales projections for this drug, but I'm only going to talk about the SWOT analysis because I do want to update those figures on the sales before sort of formally presenting it. So let's look at the strengths and weaknesses, both from a clinical and commercial perspective here. The most significant strength that Trodelvy is really meeting an unmet need for patients, and it performs much better than currently available therapies. This is also a first-in-class ADC for this population, which gives Trodelvy a tremendous first mover advantage, not just in this indication, but potentially also future expansion in their pipeline. The novel mechanism is also important because it suggests low cross-resistance with other therapies that patients might have progressed on, which is especially important knowing that TROP2 is often overexpressed in these resistant cancers. So one of the downsides in being first in class is that you often have to build out testing capabilities, in this case, pre-testing patients for TROP2 expression, similarly to how patients with HER2 positive cancers are currently tested prior to treatment. However, this does not seem to be the case for TROP2 in triple negative disease, which is another advantage. So everything here on the left kind of pertains to what I was just discussing about different pipeline opportunities and different trials going on right now. So what's important here is that, I won't share my peak sales estimate in this video, but the point is that it's well short of $21 billion. But that is the price that Gilead paid to acquire Immunomedics in October 2020. So part of this price tag can be attributed to the larger antibody drug conjugate platform and proprietary linker technology that Immunomedics has. I would argue that a significant amount of that dollar value is actually related to the Trodelvy label expansion opportunities in earlier lines of therapy. So earlier lines of therapy being one opportunity. It's also another opportunity is in other TROP2 expressing cancers, again shown on the left, and also in combination with other therapies. And so the pursuit of this large market opportunity is exactly what you see in these trials, many of which are actually supported by that original basket trial I previously described. Another strength with Trodelvy is its activity beyond that primary mechanism of action I previously described. And really, here there's 2 main points of interest. First, when Trodelvy kills these cancer cells, it's thought that the cells release cytokines which can aid the anti-tumor immune response. So in the context of combining with immunotherapy, as is currently being done in a trial looking at Keytruda plus Trodelvy versus Trodelvy alone, there's real potential in a durable treatment benefit. Second, we have something called the bystander effect that could be beneficial in dealing with heterogeneous tumors. So on the left, you'll see a depiction of a heterogeneous tumor that has both TROP2 negative and positive cells in close proximity. Now, without this bystander effect, for example, in an unconjugated anti-TROP2 antibody, you would only see eradication of the TROP2 positive cells, and the negative cells would continue with their pathology. However, since the tumor microenvironment is relatively acidic, Trodelvy's hydrolyzable linker allows for govitecan, remember the cytotoxic component of the drug, to also be released in the near vicinity, which allows it to attack both positive and negative cells and having a greater anti-tumor effect on the whole. So finally, in its 37-year history, Immunomedics has never actually brought a drug to market, and it could really benefit from the marketing power brought forward with Gilead's distribution experience, not just in its recently built up oncology portfolio, but also with its historical ability to penetrate hepatitis and HIV markets. So the most significant weakness, in my opinion, is some of the unanswered questions that were left due to trial design, which hopefully are illuminated more in the future. The pivotal trial does tell us some information, but it doesn't adequately tell us how Trodelvy works in patients with brain metastases, in Black patients, or in patients with worse performance status. It also doesn't tell us the effect on health-related quality of life, which is arguably the most important factor in patients progressing towards the end of their life. Additionally, the black box warning and monitoring of UGT mutated patients is less than ideal and could limit use or pave the way for safer therapies to take market share. The same is true for the dosing of an IV infusion twice every 3 weeks, which is comparatively inconvenient if hypothetically an effective oral therapy became available. It's also possible that because TROP2 is a novel target, significant investment in education will be required to promote use of this drug. This would be less true if this was, for example, the 8th or 9th checkpoint inhibitor to come to market. Clinicians are already familiar with checkpoint inhibitors. But despite these benefits of the Gilead acquisition, you could argue that because Gilead's been on such an aggressive acquisition and licensing campaign with 13 major oncology transactions in the last 2 years, you could actually spread the organization thin and dilute some of the focus on Trodelvy. So there's also significant regulatory risk, especially considering Immunomedics' history with complete response letters relating to manufacturing, quality control, and data integrity. Finally, related to the high cost of therapy, it's possible that international payers, such as in the UK or the European Union or abroad, they may be less willing to pay at all for this kind of expensive late-line therapy, but even more likely not to do so at the US price points. So again, I just want to emphasize, I'm going to skip over my projected US sales just because I wanted to refine them further. So here are our take-home points. First of all, I hope that today I showed that Trodelvy does represent a major breakthrough. And it's not just groundbreaking for antibody-drug conjugates or for TROP2 as a therapeutic target, but it actually represents the improvement of patient outcomes that haven't really changed substantially in decades for triple-negative metastatic breast cancer. We know that this therapy gives patients more time, and I can't emphasize enough, that's really, really hard to understate the significance that has for patients. But I also hope that I showed that there's a substantial market opportunity here for late-line therapy But really, the real opportunity for Trodelvy to be a blockbuster drug is its success in expansion trials and its overall commercial strategy. And then finally, I do want to leave on a note of relative versus absolute benefit. So as I said, Trodelvy will mean a lot to patients in giving them more time, especially relative to yesterday's standards. But I think it's really important to put yourself in the patient's shoes and understand the benefit in the absolute sense. So remember that patients with metastatic triple negative breast cancer are relatively younger than in other types of breast cancer. So maybe this patient is a mother who might not get to meet her grandchildren or see her children graduate college. And I think that is, obviously a tragedy. The survival benefit was only 5 months more than the standard of care. And I believe that 5 months is not enough. We need better therapies that really make an absolute difference in patients' lifespan while improving their quality of life. That's what I'd like to see, and that's what I'm optimistic for. And I think that's what I'm optimistic about, what good science can do for humanity. So I do have references. I'll sort of pause a little bit on each slide so that you can come back to it if you like. And I'd just like to thank you all, if you made it this far, for listening to this PowerPoint.